Okay, thank you. That's really great. But let's dig just a little bit further. For example, a lot of the products out there, they may have been isolated in some form, and then they mix this strain and that strain and another strain to have 7 strains or 10 strains or whatever. So explain just a little bit more of why the matrix that Biotics has, which is a naturally occurring, as you explained it to me, which is billions of years old, basically. I mean, these have lived in nature forever, and it withstood all of, you know, the challenges of time. And then extracted with their food source, still intact, and then cultured to, you know, to isolate it out and cultured and so forth. So say more about, say a little more about that as it relates to someone going to the lab and taking this strain off the shelf and that strain, just to clarify so that we all understand why a Probiotics Consortia is the optimal word.
Okay, so let's get back to the word symbiosis. The reason that Biotics is quite different than just a mix of supposedly good strains of probiotics is because, as you mentioned, they've gone through the test of time. They have evolved together so that the end products of one organism feed the second level of bacteria, and the second level feeds the third, and so on and so on. In other words, those symbiotic mixes are stable. They can be transferred through time after time after time. It can be cultured, and their force or their strength or the concentration of their end products remains the same. Whereas if scientists like to take things apart, you know, they like to take things apart so that they can, you know, understand them. And so what scientists typically do is they'll take a sample, let's say it's from nature, and they'll culture that sample on what's called differential media. And what that means is, you've all seen Petri plates with different types of agar and different types of nutrients that favors one organism over another. And so they can take a mix of organisms and take that mix and pull it apart and get individual strains.
So isolate it, basically.
Yes, so they're going to isolate different strains, and they're going to give those strains names over time, and they're going to find out what food that organism likes, what makes it tick, what are its end products, and then they're going to freeze dry it so that they're going to hold it in what we call suspended animation, and they're going to do the same to all those different organisms. But the issue with that is when you go to remix those organisms, they're not ending up in a symbiotic mix anymore. They are just a mix of organisms that may have potential benefit to humans, and very often do, but they are not being cultured with their buddies, so to speak. They're not being cultured with their co-isolates. And so when they get to the gastrointestinal tract, there isn't any dynamics going on that would preserve each one of those bacteria. And so what we find over time, what we know, is that when you take single strains, or you take, we're going to call them defined mixes, when you take defined mixes, we know that if you stop taking those, they are gone from the gastrointestinal tract in about a week or two. And so you're not getting the full benefit then of what's going on from a consortium. And let me be a little bit more specific. A consortium of microorganisms, or we're going to call this a natural mix of organisms that depend on each other, may secrete antioxidants, they may secrete B vitamins, they may secrete bioactive proteins, which interact with hormones. They may secrete bioactive proteins like lactoferrin, which steal iron from bad bacteria and give it to the tissues that need it. And so each level of those organisms in a symbiotic mix has a job. And they support both the organisms above and the organisms below. And what's really also important about that is that they clear out toxins. And they clear out toxins both by secreting compounds, which we know as bacteriocins. Bacteriocins are compounds which kill off bad bacteria. And our friendly symbiotic mix can not only eliminate those bad bacteria by secreting these bacteriocins, but also they can clean up the mess by getting rid of the toxins. And that's why a multi-leveled approach is so important. Many of us, myself included, have suffered from Candida albicans. And we know, for instance, that Candida can secrete up to 70 toxins. So it's very, very important to have organisms within the mix that can not only support positive changes in the gut, but can clean up the mess. I'm going to call it the mess. That means toxin release after these harmful organisms are being killed off in the gastrointestinal tract.
Oh, fantastic. So